Hello students, here we are with the next video on the topic bacteria, that is about reproduction of bacteria. In the previous videos we have discussed the two modes of reproduction, that were transformation and transduction, and in this video we'll discuss the last mode of reproduction in bacteria, and that is conjugation. So let's start the lesson. Conjugation is a process that was discovered by Ladybug and Tatum in a bacteria named E. coli. I've told you that E. coli is a gram-negative bacteria, and this is a bacteria that is found in the intestine of humans. Okay, so this is the bacteria present in intestine of our body. Then let's discuss something about E. coli. E. coli is a dimorphic species. Dimorphic species means it contains two types of cells, F positive or donor, F negative or recipient. Now what's F positive cells? The bacterial cells which contains F plasmid. Okay, we have discussed the several types of plasmids. So among that plasmids, F plasmid is called fertility factor, and the bacterial cells that contains F plasmid are known as F positive cells, and the cells which doesn't contain F plasmid, these are called F negative or recipient cells. So the basic process of conjugation is the F factor from F positive cell transmit into the F negative or recipient cells through a conjugation tube. So let's discuss what are the factors or what are the genes that are responsible for the whole process. Actually, F plasmid contains Trigenes. Tri is the short form for transfer. So it contains 25 transfer genes, out of which some are MPF genes and some are DTR genes. MPF means mating pair formation. These genes helps in extrudation of sex pilus, and they also forms secretion system with the help of which DNA will be transferred. And DTR genes means DNA transfer genes. The product of these genes helps in the processing of the DNA and make it ready to transfer. The complete process of conjugation is something like this. What happens when F positive and F negative cells come in contact with each other? Then the genes of F plasmids express themselves, and through the expression of MPF genes, sex pilus is formed. Sex pilus connects the two cells, and a secretion system is formed, and the next step is a cut is made in the single strand of F plasmid, and that cut is made by the relaxase protein, and there is a special site at which this cut will happen. That site is called ORI-T. Okay, I'm repeating, there is a special site in F plasmid that is called ORI-T, and at this site what happens? Relaxase protein makes a cut in the single strand of F plasmid. After making that cut, that single strand of F plasmid is transferred through the expression of DTR genes we have discussed. So it gets transferred through secretion system or conjugation tube that is formed between two cells. So it gets transferred from F positive cell to F negative cells, and after getting into F negative cells, it replicates itself and it forms double stranded. So at first we had one F positive cell and another F negative cell. After the process conjugation, what becomes? Both cells becomes F positive. Okay, so basically what happened is the genetic material of F positive cell get transferred to the F negative cell. So this was the process of conjugation.